Today I will be presenting an article called The Role of Tau in the Pathological Process and Clinical Expression of Huntington's Disease. Huntington's disease is a rare genetic neurodegenerative disease affecting around 30,000 Americans. Huntington's is caused by abnormal CAG repeats in the HTT gene, which then leads to overexpression of the amino acid glutamine, making it so the protein coded for by the HTT gene, the Huntington protein, will aggregate. The authors of this article were inspired by previous research done about Huntington's disease that suggests that there is quote-unquote a large set of possible genes distinct from the Huntington's disease locus itself that could modify the disease manifestation and progression, end of quote. And so, the scientific community leaves us with a lot of room to explore other genes and genetic variants that affect the development of Huntington's disease. The researchers decided to focus their research on a specific gene called the microtubule-associated protein tau gene, abbreviated MAPT. This is a gene involved in microtubule assembly and stabilization as well as some other neuronal functions. This gene has two haplotype classes, the first of which being the common H1 haplotype and the other being the H2, which is the rare of the two. Interestingly, having the H1 haplotype has been shown in previous research to be a risk factor for various neurodegenerative disorders and is associated with dementia development and Parkinson's disease. There are multiple experiments going on in the study, the first of which is that the researchers are trying to test whether there are hypophosphorylated tau aggregates in postmortem brain tissues from patients with Huntington's and are comparing these samples to postmortem brain tissue from other neurodegenerative diseases as well as from healthy controls. Next, the second part of the study involves the researchers doing the genotype to phenotype analysis of patients with Huntington's disease to analyze cognitive data and its relationship to the different haplotypes of the microtubule-associated protein tau gene. We will be focusing on this idea in the presentation as the experiment yields some fascinating and actually quite surprising results. For this presentation, I decided to focus in on Figure 4 from the article, which is titled The Effect of MAPT Haplotypes on Cognitive and Motor Decline in Patients with Huntington's Disease. This part of the experimentation drives forward the question of whether variation in form of MAPT gene affects the development and clinical expression of Huntington's disease. Before I elaborate on the three panels of this figure, there's a few key points in the study that are necessary to point out, which will hopefully make the findings I describe a little bit easier to understand. First, for the purpose of the experiment, even though we know that the H1 allele is dominant, the authors classify the H1 haplotype as solely being homozygous for the MAP gene, while the H2 haplotype refers to either being homozygous for the H2 gene or being a heterozygous carrier of it. Next, to point out some information about the study, I'll present that this had been a case study of 473 Huntington's patients who were genotyped for the H1 or H2 haplotype using a SNP known in the region. The two were demographically matched, and test monitoring cognitive and motor skills were administered once initially and then after a duration of 1 year. Let's dive into looking at the results obtained for the cognitive abilities test. What we're looking at here is a box and whisker plot, and you see two different figures, the H1 haplotype patients on the left and the H2 haplotype patients on the right. To break this down, the mean annual change in cognitive scores was negative 6.31 points over the course of a year for the H1 haplotype compared to negative 11.31 points for the H2 haplotype. This indicates a significantly higher rate of cognitive decline in H2 than H1 individuals, with the data being found by the researchers to be statistically significant. Next, we have two box and whisker plots, only now we're looking at scores on a motor skills assessment that had been done for the same subjects, again looking for change over the course of a year. However, the experimenters actually found that there was no statistically significant difference between the two mapped haplotypes and that there was no significant effect on rate of change in the motor performance. The researchers actually decided to abandon this part of this hypothesis completely and focus in on differences in cognitive scores instead. Here we have a graph comparing the number of CG repeats to the changes in cognitive scores in points per year for both haplotypes. What the researchers did is plot all the subjects they had analyzed and created lines of best fit for both haplotypes so that correlation could be assessed. And so, as we could infer from the first figure I focused in on, it was found that there was a significant correlation between these two variables for H2 carriers but not for H1 homozygotes. Overall, the information that we get from the experimentation that led to this figure is that the MAPT haplotype does indeed affect the rate of cognitive decline that individuals suffering from Huntington's will experience. Significantly, the H2 haplotype is the one we would consider as a risk factor, as this experiment found it to be statistically significant in influencing cognitive decline, while the H1 haplotype was not found to be statistically significant. Now, the reason that these findings are actually rather surprising is because, as I alluded to earlier in the presentation, when experimenters identified the tau proteins as one to study with respect to Huntington's, they had this pre-existing knowledge that the H1 haplotype was the risk haplotype for other neurogenic disorders. Overall, through analyzing postmortem Huntington's brain tissue and comparing it to both postmortem brain tissue of other neurological disorders as well as to healthy tissue, investigators found that the MAPT is a major factor of the pathological process of the Huntington's disease. We didn't explore this part too much in detail, but essentially the idea is that when tau protein aggregates and interacts in some mechanism, which the experimenters are not entirely sure of, it will interact with the normal Huntington protein aggregates that we know to be the primary and direct cause of the disorder. Also, researchers addressed the concern that tau pathology might be associated with age rather than Huntington's by analyzing patients with young onset Huntington's to prove this. The experiment we have been discussing contributes to the research by taking it to the next level and proving the clinical effect of this interaction and involvement of tau, and in that it has a specific haplotype of the MAP gene affects the rate of cognitive decline positively, in that it actually speeds up the cognitive decline systems of Huntington's. It is important to make note of just how significant this paper is to the scientific community and to the world as a whole. Essentially, there is this broad idea in the scientific community that other genes besides the HTT gene might be linked with Huntington's disease. Likewise, there is this idea that tau protein aggregation is involved with other neurological conditions in various ways. Here, the researchers are able to prove the involvement and interaction of tau protein in aggregation of Huntington's disease specifically, and furthermore prove its direct effect on clinical progression of the disease. The research suggests that with the information they have collected, further research can be done into the topic, into the mechanism specifically of the interaction described, as it is underdeveloped and poorly understood. Additionally, while we know Huntington's disease is incurable, we are now excited about this research because it gives us one more factor to potentially base therapeutic research around for Huntington's disease. On this slide, you will find my works cited, which includes all scientific resources and journals I have sourced throughout this presentation. I hope this has been enjoyable and entertaining, and I am personally excited to see how this article will shape the future. Thank you.